I really love thinking about metabolism in terms of maintaining a healthy weight and lifestyle. Now, I do enjoy a good meal and snacks, but it is important that you moderate the number of calories you take in and balance it with your exercise plan, which can be hard at times like these when we're all fatting on the couch and don't have access to the gym. In this lecture, we'll see how excess dietary molecules can get converted as fat if you're not burning them for energy in your daily basal metabolic requirements.
We'll look at some of the basic ways of thinking about fat synthesis, so the why, when, and where it happens, and then we'll look at how it happens. So you should be able to summarize the key steps and reactions needed to generate a fatty acyl chain. Allosteric and covalent modification are also going to be important methods of regulation, and we'll compare the synthesis to the breakdown pathway, that is beta oxidation that we just looked at. And this is another great example of reciprocal regulation.
The supplemental readings will go into a lot more detail than we'll cover in this lecture, so just focus on what we cover and don't worry about any details that aren't provided in these lecture slides. We've already talked about the hormonal response for carbohydrate metabolism, but fat synthesis is also going to be under the control of the anabolic hormone insulin. We saw that insulin helps with the import of glucose as well as any dietary fats or amino acids for cellular use, and all of these macromolecules will be broken down first in order to generate energy. Once the levels of ATP rise, remember it can allosterically inhibit some of the steps in glycolysis, and this will allow for any excess glucose to be stored as glycogen in muscle and liver cells. Glycogen is a limited reserve, so we can also store some of these excess carbons as fat molecules, and this is all going to occur using acetyl-CoA. As the cell generates energy, remember ATP and NADH can inhibit the citric acid cycle, and this is going to allow the acetyl-CoA to instead be used for fat synthesis. So if you're consuming an excess of dietary macromolecules, so either carbs, fats, or proteins, if your cells don't need them for energy or to create other molecules in the cell, they will get broken down to acetyl-CoA, which is going to be the precursor for fat synthesis. So dietary carbs are going to be broken down to pyruvate, which then enters into the mitochondria and is converted by PDC to form acetyl-CoA, while dietary fats are also going to be first broken down to form acetyl-CoA and beta oxidation. And you might be wondering why we break them down first, and the reason is that they will have a variety of different structures, so different lengths or degrees of saturation. And whenever we store fatty acid chains in a triglycerol molecule, they usually contain palmityl chains, so 16 zeros. So if we're going to use them in the cell, we can do that first, or we can break them down to acetyl-CoA and then resynthesize any 16-zero chains. Amino acids can also be converted to acetyl-CoA, so any extra amino acids that you don't use for protein synthesis will get used for fat synthesis. Let's look at the process of making a fatty acid chain.
You can break down fatty acid synthesis into 3 steps. The first is getting the acetyl-CoA out of the mitochondria because fat synthesis occurs in the cytoplasm. We saw that fat breakdown, or beta oxidation, occurs in the mitochondrial matrix, and remember this is a great way to regulate pathways by isolating them in different compartments so that they'll only work when you have substrates coming in. In the cytoplasm, that acetyl-CoA is going to be carboxylated to form a molecule called malonyl-CoA by an enzyme called acetyl-CoA carboxylase, and this is going to be the regulated step in fatty acyl chain synthesis. So step 2 really is the start of fat synthesis in the cytoplasm. Once malonyl-CoA is made, it can get used by fatty acid synthase to create the long saturated fatty acid chains, and these will be 16 carbons in length.
Remember, in order to get things in and out of the mitochondria, they do need to be transported across the inner mitochondrial membrane. That coenzyme A molecule was too big to cross this membrane, so when we want to import fatty acid chains, remember we were using the carnitine shuttle that imported them bound to carnitine and then recreating that acyl-CoA chain for breakdown in beta oxidation. If we want to export acetyl-CoA, and remember that acetyl group has 2 carbons, while an acyl chain will be a longer chain of carbons, the CoA on acetyl-CoA in the matrix can't be exported. Instead, we're going to convert acetyl-CoA to citrate in the first step of the citric acid cycle, and then citrate is much smaller and can be exported out of the mitochondrial matrix. So if we have pyruvate being produced in glycolysis, it'll enter into the mitochondrial matrix, be converted to acetyl-CoA by the pyruvate dehydrogenase complex, and then either used for energy, or if we have lots of energy already, the citric acid cycle will slow down and citrate will get exported. The same goes for any acetyl-CoAs that we make in beta oxidation. It can be used by the citric acid cycle first to make energy, but if we have lots of energy, citrate will be exported for fat synthesis. Once in the cytoplasm, citrate is going to be broken down by different enzymes to produce pyruvate and an acetyl-CoA that can be used for fat synthesis. And that pyruvate can then just go back into the mitochondrial matrix. This slide just summarizes stage 1. Coenzyme A is too large to cross the inner mitochondrial membrane, so instead, the citrate that's made in the first step of the citric acid cycle by citrate synthase will get exported, and then it'll get broken down to recreate acetyl-CoA and pyruvate in the cytoplasm by different enzymes, and this will require an ATP molecule. We're also going to make an NADPH molecule, which we haven't seen much before, but it is a reduced coenzyme that can be used in anabolic pathways. That pyruvate can then go back into the mitochondria, while the acetyl-CoA will get used for fat synthesis. It might seem a bit redundant using pyruvate to import carbons and then export them again, but remember the carbons are coming from different macromolecules, and the deciding factor is whether or not we're going to make ATP in the mitochondria. So the default pathway really is to make ATP. It's not until the levels of ATP and NADH rise that we slow down the enzymes of the citric acid cycle. And this will then allow for citrate to be exported in order to synthesize fatty acid chains. Once acetyl-CoA is exported into the cytoplasm, there are two enzymes that will be needed for this anabolic process. Acetyl-CoA carboxylase is going to carboxylate acetyl-CoA to make malonyl-CoA, which is an activated molecule using a CO2 group, and this will allow for the synthesis of fatty acid chains. This enzyme is going to be the committed step, so the first step in fat synthesis, and it will be regulated in order to ensure that fat synthesis is on when we have excess carbons in the cell. Acetyl-CoA carboxylase, or ACC for short, is going to use ATP, while fatty acid synthase is going to use NADPH as reducing power for this anabolic pathway. So we are going to need lots of energy to make these longer chains from acetyl-CoA. Acetyl-CoA carboxylase catalyzes a similar reaction to what we saw with pyruvate carboxylase in the gluconeogenesis lecture. It's going to use the energy in ATP to create a bond between acetyl-CoA and a CO2 molecule. And bicarbonate is going to be the substrate providing that CO2 molecule. So acetyl-CoA carboxylase is a ligase, and it's going to join the CO2 group onto the end of acetyl-CoA. And we now have an activated malonyl-CoA, which can be used for fat synthesis. Once we've made the malonyl-CoA, it can actually inhibit the import of fatty acid chains into the mitochondria. When fat synthesis is on, we don't want fat degradation to be on, so it's going to be inhibited in order to prevent this futile cycle of synthesizing and then breaking down the chains. When acetyl-CoA carboxylase makes malonyl-CoA, it can then inhibit the carnitine shuttle to prevent the import of fatty acid chains. It does this by allosterically inhibiting the first acyltransferase, so CAT1, preventing the chains from being transferred onto carnitine for import. Acetyl-CoA carboxylase is also regulated since it is the first step in fatty acid synthesis, so let's take a look at that. One of the main ways acetyl-CoA carboxylase is regulated is through the phosphorylation of the enzymes. So if you made a table of enzymes that are regulated by phosphorylation, you can add in ACC. There are two different enzymes that can phosphorylate it and shut off its activity. Protein kinase A, which we saw is activated in the catabolic signaling pathways, as well as AMP activated protein kinase. So this makes sense because if you don't have a lot of energy in the cell and all the ATP is being used up and even some of the ADP is being used producing AMP, then we would want to inhibit the first committed step of fatty acid synthesis by phosphorylating the ACC. The epinephrine and glucagon signaling pathways can then help with the breakdown molecules to make ATP for the cell. When we're in the anabolic state, following insulin signaling that demonstrates we have lots of sugars and other molecules coming into the cell, a phosphatase can take over and dephosphorylate the enzyme in order to reactivate it for fat synthesis to occur. So this circle and square are just representing the conformational changes that occur when you phosphorylate the enzyme to inactivate it and prevent the carboxylation of acetyl-CoA. So when it comes to regulating fat metabolism, acetyl-CoA carboxylase is the committed and regulated step for fat synthesis. It's inhibited when the cell needs energy by phosphorylation, either mediated by the GPCR signaling pathways, which can be initiated by glucagon or epinephrine, as well as by rising AMP levels. Dephosphorylation occurs to reactivate the enzyme when insulin signaling happens, and this will activate a phosphatase that will remove the phosphate using water. ACC can also be allosterically stimulated by citrate, and remember, citrate was the molecule that was coming out of the mitochondria in order to produce acetyl-CoA for use by ACC. So it's a form of feed-forward regulation. While the final product of fat synthesis, palmitoyl-CoA, which has 16 carbons in it, can also inhibit acetyl-CoA carboxylase allosterically. We also saw how the product of acetyl-CoA carboxylase, so malonyl-CoA, can inhibit the carnitine shuttle, and this will prevent the import of fatty acyl chains for beta oxidation, preventing that futile cycle of synthesis and then breakdown. Now that we have this activated form of malonyl-CoA, fatty acid synthase is going to use it to add carbons together to form the fatty acid chain. FAS is a very large enzyme, and it's shown on the left, where each of these letter abbreviations represents a different enzymatic activity. So each of these regions will be different active sites, and there is a part of the protein called the acyl carrier protein that's going to carry the intermediates between the different reaction sites. On the right is the structure of ACP, so it is a protein component, and you can see some helices that are contributing to its structure, but there's also a covalently bound cofactor. This cofactor is called phosphopanthenine, and it is a vitamin B5 derivative, and it's got the same structure that we see in coenzyme A, which is shown as well. This yellow circle is actually the sulfur atom where we attached either the malonyl or the acetyl group. So fatty acid synthase is going to transfer these acyl groups from coenzyme A onto the acyl carrier protein. You can kind of see how this long phosphopanthenine group can be quite flexible. There's rotation around all of these bonds, so this really allows for the intermediates in the reaction to go from one active site to another. And there will be 4 key reactions in order to build up a fatty acid chain. So let's look at this mechanism. Fatty acid synthase kind of has an opposite mechanism to beta oxidation, or the breakdown of fatty acid chains. There will be rounds of 4 different enzymatic steps shown on the right. The very first round, and only this round, is going to start with an acetyl-CoA molecule, as well as a malonyl-CoA molecule from acetyl-CoA carboxylase. The carbons from these molecules will be transferred onto acyl carrier proteins, and the first step is going to involve a condensation reaction where the decarboxylation of the malonyl group is going to drive the formation of a new bond between the 2 carbons of malonyl with the 2 carbons of the acetyl group. So a single acetyl group, shown in blue, is needed in the first round of fatty acid synthesis, and this is going to be the omega end of the fatty acid chain. The enzyme that catalyzes this reaction is called ketoacyl synthase, or KS for short, but you don't need to memorize these names. The next 3 reactions are going to help get the chain in a reduced form. So there's going to be 2 redox reactions using NADPH as reducing power, first to reduce the ketone to a hydroxyl, and then we'll lose water to create a double bond, and then another redox reaction to get rid of the double bond and create the saturated chain. And remember, each of these 4 reactions are taking place in a different active site, and it's the acyl carrier protein that's moving each of these intermediate metabolites between the different sites. The chain now has 4 carbons, 2 from acetyl-CoA at the end, and then the 2 pink carbons from malonyl-CoA. And we can then repeat these steps using a second malonyl-CoA. So starting at the top, our 4-carbon butyryl ACP will replace acetyl ACP, and a new malonyl ACP will have a CO2 molecule on it to allow for the decarboxylation and the condensation of the 2 carbons from the next malonyl-CoA onto the growing chain. We'll then get rid of the ketone group that was on butyryl-CoA to form a saturated chain. If we compare fatty acid synthesis to beta oxidation, fatty acid synthesis is going to take place in the cytoplasm, while beta oxidation takes place in the mitochondria. Synthesis is an anabolic process, so we are going to require energy, and fatty acid synthase is going to need the reduced coenzyme NADPH, while in beta oxidation we saw NAD plus and FAD being used to make NADH and FADH2 in 2 redox reactions. The synthesis is going to require a single acetyl-CoA as a substrate and then multiple malonyl-CoAs that are going to provide 2 carbons in the chain for each round, while each round of beta oxidation made an acetyl-CoA, with the very last round producing 1 extra acetyl-CoA. So the number of rounds for a certain chain of length will be similar if you look at fatty acid synthesis versus beta oxidation. The enzymes, however, are quite different, so we don't share any of the enzymes like we saw in glycolysis and gluconeogenesis, and the stereochemistry of the intermediates is also going to differ. The conformation of that hydroxyl group on the beta carbon is different, so say for some reason one of the intermediates was released mid-pathway, you wouldn't be able to use it in the other pathway. So it's really neat how the cell has similarities but very obvious differences between its anabolic and catabolic pathways. In order to create the fatty acid chains, we do need both acetyl-CoA carboxylase and fatty acid synthase working. Fatty acid synthase will run 7 times in order to generate a 16-carbon chain, and it's going to involve a single acetyl-CoA in the first round, and then each round after that will use malonyl-CoA to add 2 carbons for a total of 7 malonyl-CoAs. This is an anabolic process, so we're going to need a lot of ATP, 7 for acetyl-CoA carboxylase, as well as 14 NADPHs for fatty acid synthase in order to create that 16-carbon chain. And once the 16-carbon chain is released, it can be elongated to longer chains, but this is going to occur by different enzymes in the ER. And we're also going to use malonyl-CoA and NADPH for these reactions. We can also add in double bonds, and this will be mediated by desaturases, and this will also require NADPH and oxygen. The last thing I want to mention is how you actually store these carbon chains as triacylglycerol molecules or triglycerides. The starting molecule is actually going to be a phospholipid called phosphatidate. So you would use a membrane phospholipid and first remove the phosphate head group to create diacylglycerol, and then an acyl transferase is going to add on that third fatty acid tail to make the triacylglycerol molecule. Insulin signaling can also help activate the enzymes needed to transfer that third tail on, in addition to dephosphorylating and activating acetyl-CoA carboxylase that we just saw. But it can also promote the production of fatty acid synthase. All cells do have the ability to make fatty acid chains for use in their membranes, but your liver really is one of the main tissues responsible for fat synthesis. You may have heard of fatty liver disease when there's a defect in the export of fat molecules and they start to build up. But usually your liver will export triglycerides in large complexes called lipoproteins, which are really a mixture of lipids and proteins and help with their transport. And we'll look at their structure in the next lecture. So just in summary, we can store excess carbons from carbohydrates, fats, or proteins as saturated fatty acid tails in a triacylglycerol molecule. The synthesis of fats will occur in the cytoplasm, so we do need to first export acetyl-CoA as citrate since that coenzyme A molecule can't cross the inner mitochondrial membrane. And it's acetyl-CoA carboxylase that's going to commit the carbons in acetyl-CoA for fat synthesis by adding a CO2 molecule to form malonyl-CoA. Because it is the committed step, it is going to be regulated primarily through phosphorylation, but also allosterically by substrates and products. And then fatty acid synthase is going to use the decarboxylation of the malonyl group to build up the chain, adding 2 carbons in each round and using reducing power in NADPH to produce the saturated chain. This will make a 16-carbon chain, which can then be elongated or modified in the endoplasmic reticulum. So just to put this all into context, I want to leave you with a thinker question. There are a lot of different types of diets out there, and now that you know a bit about fat synthesis and biochemistry, what's the biochemical basis of a low-carb diet, and is it really any better than any other kind of diet? Each diet has its pros and cons, and they've all been developed for a variety of reasons, and hopefully by the end of this course, you'll appreciate the importance of moderation and supplying your cells with all the different molecules they need for all the different reactions that take place. Check out the BBQs posted on Quercus before moving on.